Thank you, first of all, for this amazing opportunity to be here at ESI to talk about flexible minds. I'm a computational systems neuroscientist that really wants to bridge this gap between neuroscience, ecology, and evolution of social behaviors. Most of what we know today about the brains of animals comes from the study of inbred animals, inbred species in very artificial conditions. And I want to use integrated biology and cutting-edge technology to bridge this conceptual gap. So today I'm going to talk a selection on my past work and some of the things I'm doing in the present and future. So first, let me start by acknowledging that the field of animal social ethology is a very rich field that has taught us a lot of things about the fascinating lives of animals and their interactions. However, unlike in many centuries ago, the observation of natural behavior is no longer restricted to what either natural historians do when they go to the field or what neurophysiologists do with their inbred lines in the laboratory. Nowadays, we have, like, as Yossi showed, some fantastic technologies to follow wildlife. We also have everybody with a phone camera in their pocket. And this has created an amazing gap between how society views animals and how science has viewed animals for the past centuries. And this gap, to me, is about flexibility. So to you, it might be about whether you have a predicate function or model-based, or if you have shortcuts, or if you have seen a lot of anecdotes. So I'm going to start by showing you some anecdotes. Some of them started early. First of all, this here is a dog that is letting itself being pinned by another dog. This is called, you can see, like, this little dog would never pin this other dog. This goat would never push a bull. Now you will see this orangutan respond to a magic trick. And here you will see this turtle helping another turtle turn around. And here you will see a dog trying to rescue a dog that fell in a pool. And the access that we had to this many behaviors, this many what I call flexible behaviors, is amazing. And I think this is a gap that we really need to bridge and we really need to explain. So this is the main question in neurobiology that I'm interested in. How do complex and flexible social behaviors arise? And I want to posit today that one such lens to see flexible behavior is to study play behavior. Play behavior can be our lens into flexible behavior in two ways. First, play could be a complex and flexible behavior in itself. The second way is that play could be the evolved drive to develop flexible behaviors in adulthood. So I'm going to start by doing a brief introduction about what play is. So first, I would say that play, it's older than culture. This is the beginning of the book Homo Ludens, and it says that because animals have not waited for humans to develop forms of play, we know that many taxa exhibit play. So for example, they exhibit locomotive play, which is figuring out how to move their own body and how to do things sometimes wrongly. And sometimes that slowly starts to interface with objects in the environment. And that's what we call object play, trying to figure out and make a model of the objects and the properties and the affordances that the objects have in the environment. And thirdly, we have what is called social play, and is trying to figure out how animate things in the environment, like other animals, can move and how we can connect with them and how we can learn from them. So here a lemur is learning that it can grab the horns of the deer, and the deer is learning that it can push the lemur into space with its head. So they're both trying to learning how to interact with each other. And this makes play this amazing thing that bridges us with other animals, and that allows a dog to play with a cat, us to play with a cat, a dog to play with us. So humans do this too, and it's incredibly important for their development. The social and cognitive development of humans requires that the humans play in childhood. In fact, playing is one of the rights of the child by the UN Charter that studied this. So I want to just say that as a way of framing this, since everybody has brought up older thinkers, that one way to think about play is to think about affordances. So we can think about social play as the aligning of affordances. So what is affordances as the concept by Eleanor and James Jerome Gibson is the quality or property of an object that defines its possible uses or makes clear how it can or should be used. And for example, you can have three cases here. You know that a button is something you should push. It has the affordance of being pushed. A switch has the affordance of being flipped, and a knob has the affordance of being rotated. But already Gibson was saying that all social behaviors, in a way, depend on perceiving what other people can do and what you can do for other people. So in these affordances as well. So one way of thinking about it is that depending on who you meet, you might want to give them a fist bump, or you might want to shake your hand. There's some people that you send them an email and you know you need to send them an email back 2 weeks, but there's people that you should wait a bit longer. So there's different ways. So you learn what are the affordances, what you can do with others, and you build a model of them. And I think this is what play is doing, and this is what social play is about building these models. But play has also not transcended the primary forms that animals show, like locomotive play in this case with my nephew, but gone into different forms of rules, roles, and effectors. So in the talk today, I will talk to you about play as a complex and flexible behavior in itself, and I will try to answer two questions using a hide and seek as a model. Are rats capable of playing games with roles and rules? And how does the brain represent flexible play during hide and seek? So, and then in the second part, I will talk to you about current work and future work about the evolution and ecology of flexible minds. So when I was doing my PhD, at the end of my PhD with Michael Brecht in Berlin, we asked ourselves this very ludicrous question: can rats play hide and seek? And we set to study this with Annika Reinhold and Konstantin Hartmann, and this is the paradigm that we used. So we had a rat that was familiarized with a human, in this case, Annika, and the rat would jump into a start box. Then one of two possible things could happen. In seeking trials, the box would be closed, then the experimenter would go hide, then the rat would jump out after the box is opened, find the experimenter, interact by form of tickling and hand chasing and different forms of play with the experimenter. And this was the only reward that was used to teach the animal how to play this game. And then the rat would be returned to the starting point for a new trial. And we did blocks of trials. And then in a hide trial, the only difference is that the box stays open and the experimenter stands next to the box counting. And then the rat knows it needs to go out of the box, now go far away from the experimenter, hide, be found. There's the interaction that is the only rewarding part, and then it returns to the start of the game. So this was a ludicrous idea in 2019, and we wanted to do it in the biggest environment that we could. So we went into our basement of the BCCN and basically set up this whole system to track the animal and to let it roam in a 30 square meter room, and to track the behavior in a very flexible way, where most of the time the animal is in control of the behavior. So here you can see different trials of seek, and you can see how the trials are very different in time. These are not just like the trials where you present something, then 1 second later something happens, then another second later something happens. These trials can last 50 seconds to 250 seconds, depending on what the animal does. And in most cases, either the experimenter is hidden or the experimenter is counting. So most of the behavior happening is actually up to the freedom of the rat. So I point that out so that you are primed to what will come in later in the talk. So let me show you how this looks like. So how does random seeking look like? So this is a first trial that you will see. This will be a shorter trial. So Annika puts the rat into the start box. Then she goes hide. And then this will be a quick trial. So the rat will find Annika pretty quick. You'll see the box opens by Annika's control here. And then you will see the rat peep out. And then at some point, it will jump out. It will do a little trajectory. And then it will find Annika and do like a beeline towards Annika. So it finds Annika. And then there's this moment of interaction that includes tickling, includes different forms of cuddling. And it also includes like playing with the hand. You will see like a moment where there is some hand chasing. You'll see that the rat likes following around the hand. And this is the only form of reward that was used to train completely the rat and teach the rat how to play this game. And then Annika returns the rat to the starting point, and here we go again. Now I'll show you a more extensive trial sped up, and you will see the rat jumping out. And in this case, it doesn't find Annika quickly, and you will see it will go through many different places in the environment at its own pace, and search through several places at the same time, awkward places. And then at some point, you'll see like it sees Annika and it does a beeline, goes straight to her and finds her. So how do we know the rat was not just randomly searching and was actually trying to search for the experimenter? So we performed 3 different tasks, one in which the experimenter would hide randomly, one where the experimenter would let the rat cheat and see in which direction it was going to hide, and one in which the experimenter would hide 5 times in a row in the same place, eliciting some kind of short-term memory, and then a rebound place in the 6th trial. And as you can see by the search times depicted here, the animal is capable of using vision as a cue to know where to go to the experimenter, and is trying to go fast to the experimenter, and also uses memory to also try to go fast towards the experimenter. So this shows that the rats develop flexible behaviors to try to seek for the experimenters, and you can use different types of cues and different sources of information. So how does hiding look like? So this is how hiding look like. This is one trial. So Annika puts the rat in the cage. Now the box stays open and Annika starts counting. And you'll see that at some point the rat realizes this is not a seeking trial. So it jumps out, goes for a place, and just hides. And now Annika will try to find the rat and will pretend like it doesn't know where the rat is. She obviously knows where the rat is. We made it very easy for the human. And you'll see that even when Annika passes, the rat peeps out and goes like, oh, where is she? Oh, there she is. Okay. And then comes in, and then there's, again, this social interaction, and all the different hiding places were equally rewarded by Annika, even if the rat hid behind this occlusions, behind the transparent, opaque boxes, or in different corners of the environment. And here's another trial. I want you to see the diversity of behaviors, because one of the things that I want to show is about the flexibility. So it does a partial hide, then it comes and hides here, then Annika finds it, and it goes and hides again. So this is just to show that we created that assortment of like YouTube videos of the rat doing interesting, amazing behaviors. So how did we look at this? So we studied a bit like what determines where the rat wants to hide. And we noticed that the rat is more propense to hide behind opaque boxes and cardboard covers than transparent enclosures. And you might think, okay, that's because like they hate light and rats have an aversion to light. These rats definitely do not. They're very happy being in the basement. But also we tested that. First of all, we looked at how frequently they entered these boxes, both the transparent and opaque boxes, during hide trials and seek trials. And you can see that they enter the boxes much more frequently when they know that it's a hiding trial, when their role is to hide, than when they are in a seeking role. So that implies some kind of change in the contextual change in their behavior with respect to these boxes. And secondly, their preference for the opaque boxes only expresses during hide. It doesn't express when they are in a seek. In all these cases, when they enter a seek, they just choose equally between the opaque and the non-opaque boxes. So this shows us that rats develop kind of flexible behaviors that express in some level or anecdotal level of understanding of the game that hiding is to not be seen. So I started by showing you all these YouTube videos and all these videos of animals doing amazing things and how we have captured them. And I wanted to show you a couple that we have found. For example, this is an example of an animal rehiding that you already saw one brief example before. But this one here, now it has a logger in its head, and you'll see, like, we record neural activity in a second. And you'll see that Annika here now is trying to pretend that it's having a hard time finding the rat. And you'll see how the rat behaves. So Annika is just moving around, and the rat is keeping an eye. So, okay, she went there, so I can come here. Okay. So she's crossing there, so I can cross to the other side. And okay, wait, she's coming. Okay. So you'll see like the different behaviors. You'll see like she crosses again now in a second. You see the different behaviors that we found that the rat was trying to perform while still playing this game. And this was, so it went and hit again and came back. So the animal in some way started to like control its own play, its own hide and seek kind of game, and playing with us instead of us playing with it. This is another example. This is Annika finishing completely the experiment and going to like turn off the system, and the rat is like, "Where are you going?" I want to start a new trial. And we saw a lot of these kind of behaviors of the rats trying to self-initiate the game in itself. So one of the other things that we really looked at that we were very interested in is in their vocal behaviors, because there is an association between play and vocal behaviors. This is exemplified here from Ishiyama and Brecht. I don't know, I hope the audio comes through in this. So this is a rat that will be tickled, and you can see it produces this ultrasonic vocalizations when it's tickled, and it really loves the hand. You'll see it's like, where's my hand? And we wanted to measure like this kind of vocalizations in this play paradigm in which the animal is also interacted with, because we think like this was the rewarding part of the game. And basically, we found that after interaction, we covered this array, this place with ultrasonic microphones, and we found that after the interaction, there's an increase in both roles. The seek is in magenta and hide is in green. Both roles, there's an increase in the call rate of the rats after the interaction starts. But this was something we expected because there's tickling, there's hand chasing. But what we did not expect is that this would translate into other parts of the game, that the rat would now start calling in other phases of the game. For example, when it jumps into the start box to start a new trial, when it jumps out of the start box to start a new trial and start either going to find a place to hide. And also we found a particularly interesting difference when the animal is finding the experimenter in magenta and being found in green. So when it's being found, it's silent. When it's finding the experimenter, the moment it sees the experimenter, and in the moments it starts that beeline, it vocalizes. So this is something that we found really interesting because the rat is repurposing some of its natural behavior for parts of the game. And this is something we could not control because this is ultrasonic, so there was no way to directly reinforce this behavior of the rat. So we could not immediately reward it and tell it like, oh, great, you vocalized, okay, do it again. So we found it a very interesting phenomenon. And now let me switch into what we did neurally. As I told you, these rats had a logger that we got from Deuteron that was implanted into the prefrontal cortex, an area that is related to decision-making, to also to social interactions, and to many things that were mentioned today, and creation of models and creation of rules. So this is an example of a neuron recorded in MPFT, in the infralimbic cortex. And you see that this neuron gets active when the box is closed. And you will see that at some point the box is opened remotely. And the neuron becomes silent. And if we look at the activity of this neuron over time, we can see that the peaks of activity kind of align with the different notches here that are the beginning of the trials of the SyNC behavior. So we could count basically how many trials the rat was doing by looking at this single neuron that was responding positively to the closure of the box and negatively to the opening of the box. But beyond this, we looked at the activity of many neurons, and in this case, in order to look at it in a compact way to have the average activity of each neuron, we had to do something quite gruesome, which was to align every single trial to the same kind of time frame. So we had to time warp all the trials for each neuron to the same time frame, to the median times of a trial. And this required stretching of the neural activity in all different ways so that everything could be aligned. When, as you saw in the beginning, all the trials are completely different, are very different one from another in their duration and the different parts of the trial. But even after doing this, we found that different neurons that are depicted here as rows and are clustered using a hierarchical clustering algorithm are responding to different phases of the game. So for example, this orange cluster is responding to the jump out when the rat is about to jump out. And this cluster here, this yellow cluster, is more active when the rat is being returned into the start box. This is the cluster, the black cluster here is when the animal is in the start box, and so on and so forth. So even with the average activity, we could recover like some sequential activity of the different states of the game of hide and seek. But we did this in a way that I like to call direct physiology, which was to align brain activity to the behavior of the animal. But there's a different way of doing this, which is to do reverse physiology, which is to align the behavior of the animal to the neural activity. And this is a very interesting thing to do because this is very advantageous for flexible and complex behaviors in which we don't know when the stimuli is going to happen, what's the important part for the animal, etc. So we can count on the brain telling us what is the important thing that the animal is paying attention to. And by reversing the arrow of inquiry, what we can decrease is like our own experimental bias of saying what are the things we want to align and what are the things we want to study about the activity of this brain by looking first at the activity and seeing what things in the behavior correlate with that. So with that, gives us to this other paper that we worked on how the brain represents flexible play during hide and seek using a completely different approach. So basically what we did with the hide and seek paradigm is to do unsupervised state discovery. So we have the activity of the PFC, and first we lower the dimensionality, and then we use a hidden Markov model that what it does is basically take these low dimensional factors and basically tell us when there are different discrete states that are transitioning in time as the rat performs its behavior. So this is unsupervised because the algorithm knows nothing about what behavior is going on. It's just looking at the neural data and segmenting neural data in time to see what happens. And then we can look at these states and see what behaviors this relates to. Just to explain a little bit what hidden Markov models are, you can think about like this. Let's say you're in an indoor building and there's a weather going on outside and you don't know what the weather is. So that weather becomes non-observable, but it has certain probabilities and it also has certain transition probabilities. But there are some things that are observable. For example, let's say you're in a casino. Casinos don't like you to see how it is outside because they want you to stay there forever. Okay, so but you can see how people enter the casino if they enter with sunglasses, with umbrellas, with mittens. And according to these 3 lines of observation, you can try to infer what is the probability that the outside is in a given state. So you can do this by using hidden Markov models. And in our case, those observations are the neurons, which in reality, in the end up being the factors. So you can use the different factors as different independent observations to run a hidden Markov model that would find different states, which are these colored areas here. And you can see that in this single trial, the color of the state that is found is changing with the different phases of the game of hide and seek. But this is just one trial. And I showed you before that the trials can be very flexible from one to the other. And you can see here how even though the trials are very different and there's such flexibility, that the different states are still being found in the neural activity, even though the model has no idea about what behavior is happening when. And it becomes even more clear once you do the same trick we did before with time warping the neural activity. Now we time warp the states, we put them all together, and you can see how the different states are clearly appearing at different moments of the hide and seek phase. And this is for other sessions. But we didn't want to stop there, because one of the advantages of doing this reverse physiology is that we can go through states that we know nothing about and try to inquire what those states of the brain are representing, regardless of what paradigm we were studying. So this is akin to looking at like how before we do direct physiology. So we compare the activity of a neuron to a certain stimuli that is happening here. And reverse physiology would be like doing a spike trigger average. So we want to know for each spike, what are the certain stimuli that are generating that spike. In this case, we want to know for each state, what are the behaviors that are appearing during that state. So then what we did was to create these collections of videos for each of the states that our algorithm found unsupervisedly. So this is an example here of what I call a transit state. And you will see that all iterations of this state that appear are when Annika takes the rat back to the starting point. That was one iteration of the state. This is another iteration of the state. So this is one of the states that we presumed something could be happening because it's one of the phases of hide and seek. And one way of looking at it in a compact way, these video collections, is to just do a superposition of all these frames, removing the background, looking at what changes from the background, and then superimposing all of them. And then you can see basically this is depictions of Annika just coming back to the start box. But if we do it for something that we don't know what it is, for example, like this, we can see that this state that was predominantly active after the rat jumped out is related to when the rat is running. So this is a state that is unrelated specifically to a phase of hide and seek, but it is related more specifically to when the rat is running. Now, this is a state that is related only to when the rat has a wall on its right. And we never would have studied that there was a state to a wall on its right. We also found states that correspond when the rat is peeping out of boxes. We found states that looked sometimes that the rat was confused. So there are lots of things that we could find by using this approach that we never would have in principle studied. So I think reverse physiology allows us to find brain behavior correlations that go beyond some of our experimental bias. So at the beginning, I showed you a bunch of flexible behaviors of and kind of strange-like behaviors of this self-handicapping that this cow is expressing towards this goat. And we found that play is a great tool to look at these flexible behaviors, and it's a flexible behavior in itself. The animals use flexible information and use flexible cues. They express game understanding. They repurpose the vocalizations as part of the game. They do trial self-initiation, and they have complex rehiding behavior, like this YouTube-like video that I showed you today of the rat just like going to the opposite place where Annika would go. So we think that this expresses some way that the capacity for role gameplay and to have models of each other and of each other's roles may be evolutionary old. So, and I also showed you that when we found that the prefrontal cortex can represent the brain, like a brain-based ethogram of the behavior that is going on by the animal, and that we can use reverse physiology to lead us to what were the behaviors that were triggering the animal's brain to go in certain different states. So I want to continue to what I will do in the future. And for that, I want to just discuss what we learned and what we didn't learn in this study. So I think what we learned is that play is a complex and flexible behavior in itself. What we didn't learn is what happens about the evolution and the ecology of flexible minds and the evolution and ecology of play in itself. So what drives animals to play from an evolutionary standpoint and from a mechanistic perspective, and how do flexible social behaviors are affected dynamically by animals that are actually living in an environment where we see them all the time. So I want to talk to you briefly about two directions that I'm taking now at Harvard and possibly in the future. And one of them, both are trying to answer also this question: how do complex and flexible social behaviors arise? The first one is about how does social play features evolve in wild deer mice? And the other one is how do brains orchestrate social behavior in the wild? So let me talk to you about this one. So first, like, deer mice is a New World rodent that lives in North America that has conquered all of North America. And there are up to like 50 different species from Alaska to Central America. They live in forests, they live in Alaska, they live in Florida, and there's many different species. So this allows us an access to the whole genus. And this generally is very useful because they have behaviors that can be genetically tractable that allows us to find loci that are related to the expression of these behaviors, and behaviors that are very comparable and that either have like been conserved or have diverged in the genus. So more specifically, one of the questions I asked myself when I came to Hopi Hoekstra's lab was, I really wanted to know what's going on with play in this collection of mice. I thought it would be like a unique opportunity to see what is going on. So one thing we knew, and I knew before joining the lab, was that permiscuous mice play. You can see they do kind of this rough and tumble play that rats do, and that dogs do, and that kids do. But one of the interesting things, if you look at these old papers, is that rats play at a very high complexity. Mus musculus plays at a very low complexity. And honestly, I don't know if they really play. But when I saw that Permiscus maniculatus, which is one species of Permiscus, was right in the middle, I was like, this is an opportunity for variation, right? We have 50 different kinds of species. Maybe we can see variation in this play behavior. And then I saw that Microtus, which is voles, different species of Microtus have some variation in this play behavior. So then I decided to look at different species and subspecies of Permiscus and try to capture their play behavior. So this is work that was started with Emory Sabatini and Shadev McFadden at Harvard. And we took these different populations of Permiscus mice that come from different parts of North America. And we did this experiment where weaned mice of weaning age are first separated together as siblings of the same sex. Then 24 hours later, they go through 24 hours of isolation living alone. And they are, in those 24 hours, they are exposed twice to this pink box, which is a play arena, in which they will be reunited 24 hours later for 30 minutes to play. This is a classic play reunion paradigm that is sought to elicit play behavior when the animals are reunited. And in this kind of conditions, using this Raspberry Pis, we were able to record video and audio of the animals doing this behavior. And this is, as far as I know, one of the first videos, actual videos of Permiscus and recordings of sound of Permiscus actually playing. So I'll show you. What I want to point out first is that if you look at the spectrogram here, this is frequency and this is time. You see this red box. There's all these little dashes of lines that are all the vocalizations that the mice is doing. And I will put a red box in the video when this moment starts to happen. And you will see what the animals are doing when that happens. So at first, not much is going on. There's not many vocalizations. And then suddenly they start to do this rough and tumble play where they kind of do a little bit of boxing, a little bit of wrestling. And then at some point it's done and they're still friends. It's all good. They keep going. So this is not fighting. I think I removed the slide. I don't know why, but when Permiscus fight, it's like aggressive. They're hitting all the walls and they're just dropping on top of each other and pinning each other down for like minutes. But let me tell you that one of the first things we analyzed when we looked at these videos was these vocalizations that we know in rodents are generally associated with appetitive behaviors and very specifically associated with play and with tickling. So if you look at the vocalizations of mice normalized by all the pairs, normalized by the amount of mice in the box, obviously, and you look at the number of vocalizations, you see that in the first exposure and the second exposure, they vocalize very little. So the vocalizations they're doing, they don't do it too much when they're alone, but once they are together and reunited, they vocalize a lot. And this peters off as the 30 minutes go by. If we look at it through all the different animals of the different species, color-coded, and also all the different pairs, once they're reunited, you will see that there's this increase in vocalizations, but you will also start to see that it's particularly predominant for some species over other species. So this is a compacted figure showing the same. So this is the numbers of whistles that we call the USVs divided by the mice, divided by the seconds. And you can see that that all of them kind of have this increase, but some of them have this larger vocalization when they're reunited. And basically, this is just that last panel blown up. And you can see that some species have a lot more vocalization during this reunion phase. And some other species of Peromyscus have very low vocalizations during this reunion phase. And Mus has very low vocalizations during this reunion phase. So we have 271,557 vocalizations. And what you generally do with that is first to try to study that feature space. And we know we can segment these vocalizations and then plot them into feature space based on their spectrograms. And some of the variables of the vocalizations map nicely to this UMAP space. And especially one of the things that we're excited about is that the different species that we study can also be mapped into this UMAP feature space based on the spectrograms of the individual vocalizations. But I want to quickly, more interestingly, show you that some of these vocalizations are, like, as I pre-announced, related to play behavior. So one of the things we're doing, we're using dipethogram, which is a supervised clustering algorithm for behavior, to cluster pro-social, to make a pro-social ethogram that includes play, chasing, huddling. Huddling is when they're close together. Allo-grooming, which is when they groom each other. And you can see that there's like increases in vocalizations that are related to play. And if we look at two species, one that vocalizes very little and one that vocalizes a lot, you see the difference in the appearance of like play behavior in this 30 minutes of reunion. So overall, like, this is just a very rude look at this. There's a lot more going on, but just to exemplify very fast, you can see that even though huddling and grooming, the proportion of the expression of that behavior in the 30 minutes is not positively correlated with the total vocalizations, we can see that for play, this is true. The more play there is, the more vocalizations there are, and there are many different ways of showing this. So in the future, what we want to look at is at many different things. We want to do, for example, cross-fostering studies to see if the play behavior is genetically heritable, or if it's transmitted, as Yossi showed today, by development, by interaction with the parents, or by interaction with the siblings. And we want to do interspecies play. And we also want to do physiology. So I've been working in the past few years to establish techniques for doing recordings of dopamine using GrabDA dopamine sensors. In this case, you can see this mouse that has an optic fiber, and we're doing phytophotometry to record dopamine in what is called the tail of the striatum, or the posterior striatum, which is less related to reward, but more related to threat and to the finding novel objects in the environment. And you'll see repeated times where the animal is either looking or getting close to the object, and you see that dopamine we can see this expression of this peak in dopamine. And also, I've been collaborating with Northwestern University. with a group that is developing fiber photometry, wireless fiber photometry, so that we can do this in like playing mice and interacting mice. And even we can do it in enclosed spaces like their burrows, which they love to live in. And finally, so in this, I want to answer a couple of questions. What are the neural bases of play variation in deer mice? And I want to also try to ask, how does play affect the development of social and cognitive behaviors in deer mice? Because we know, like in humans and also in rats, that play affects how you develop your cognition and your social cognition. But Peromyscus has incredible advantages to study this because they're interfertile, so we can do genetic crosses of them and separate different features of play and seeing which features affect more the cognitive development of the mice. So the last chapter and the last thing I want to tell you about is my dream, is what I want to do. And like coming after the amazing things that Yossi has shown you is so inspiring to try to do things like that. And one of the things I think about a lot is the interaction with the environment and the animals, and how we should study animals that are doing their natural behaviors, like Yossi has shown you. And so I am trying to develop a new model to study the brain in a system that allows us for natural behaviors. And this new model, I think, is the agouti, which is a champion species for social behavior and many other things that I will show you in a second. And it solves a lot of challenges for rat and mice models for being studied in the wild. First, they're very big. They are 3 to 6 kilograms. They're diurnal. You can look at them with cameras during the day. They don't hide in burrows. They don't fly. They don't climb trees. You don't need to do 3D mapping. So there's a lot of advantages. They also have this very nice large brain in comparison to the brain of rats and mice that even has this little gyra here. And they express a lot of fascinating behaviors, like a lot of agonistic behaviors that includes erectile hair, jousting. They also have incredible sexual behaviors that include, for example, urination, in which the male urinates on the female and the female does a frenzy dance. But they are most famous in natural history for doing this behavior called scatter hoarding, in which they hide seeds underground. And even natural historians have described that they put leaves on top of their caches. And when I saw this, I could not believe it. I thought like, oh, that's a nice story. But there's actually videos and many videos of them doing this. And they are really famous for this because they have saved a lot of the megaflora. Once the megafauna in Central and South America went extinct, they took that niche and saved all the seeds and put them in the ground and made the megaflora be sustained until today, because they scatter hoard all these seeds, putting value for the future in different places of space. So how do you start a new animal model? You go and do a little meet and greet. So I've been to Panama to seeing where they are. And Yossi, who has collaborated with Paige, maybe you've been there. Like, it's amazing. They're everywhere. They are in the town. They're not very skittish. So I would this is me in my Airbnb, just sitting there, like giving them nuts and crackers. And I even got to hand feed them. And I also went to the zoo.
We got more.
These behaviors are retained. And you see, like, we gave like 5 nuts to this agouti. And the first thing it did was to go back to his home cage, like do a little hole. At some point, we'll see it in the video. Does a little hole with its paws. It tastes the thing a bit. What is it? Okay. Then hides it. And then just comes, and we gave it like 5 things in a row in an animal that hasn't seen the wild since it was a baby, and it still expresses this beautiful behavior. And they are like very amenable to humans. Like, this is me in Panama, and you will see like it's a bit like there's a little bit of neophobia, but you will see like it's coming and just grabbing this piece of cracker from my fingers. And also, it's very interesting because they do this kind of chuckle when they grab food. They have this like celebratory vocalization. So that's basically what I wanted to tell you. I'm also working with OpenEFIS to develop a system for logging them that will allow us to do like state-of-the-art wireless multi-area recordings, like 512 channels with onboard camera and microphones to study their behavior from an egocentric point of view, accelerometers, and will allow us to work with freely interacting animals for up to 8 hours. We're developing new ways of using batteries to do this. And what I want to do is multi-animal social behavior that has allocentric and egocentric behavioral recording. And we can use reverse physiology to study how multi-area brain states are formed and are shaped by the behaviors that the animal expresses, and be able to synchronize the brains of animals to the wild and study their naturally complex ethograms, as I showed you before, to this diversity of fruits with this multi-area brain recordings at the same time. And I won't take more of your attention, and just to show a little, finish with a little meme that came out of our work. I'm now going to take care of my introverted friends, and after interacting with you, I'm going to return you to your hosts and thank all the people that work with me in this study. So thank you.